Folks, we want to welcome you to 2-6, a laboratory on chromatography. As always in laboratories, it's encouraged that you wear safety goggles to protect your eyes. In this lab, they'll be separating, it's a simulation for them separating colors based on the process known as chromatography. We don't want any of those colors getting in your eyes. If the experiment goes poorly or whatever, we don't want your Chromebook screen to explode and send glass and stuff into your eyes. So safety goggles are always a good idea, as well as wearing clothes that it's okay if you get chemicals or whatever on them. We don't want your clothes to get damaged. Or you can wear a lab apron or safety jacket or something like that to keep that stuff off your clothes in the event that this lab simulation goes wrong. Today's lab is one of the many ways of separating mixtures. Remember, mixtures are when things are placed together and nothing happens chemically. They're just placed together. Many colors are mixtures. We know that certain colors are made by mixing some of your primary colors, and the result is a different color that you see. And a process for separating mixtures similar to this is called chromatography. Chromatography. And as you can see here on our lab report, chromatography involves the word chrome, which is color, and tography, which is write. So this is really meaning writing with colors. Now, in this particular lab, they're going to be separating the pigments that make up leaves. Leaves have different organisms in the leaf, different cells or whatever in the leaf that are involved in the process of photosynthesis. And those particular cells in the leaves, the energy generating cells in those leaves, give the plants leaves the colors that they have. And in this lab, we're going to separate the colors within a leaf. Now, the way chromatography works is it takes advantage of the different physical properties that the colors or pigments have. Every molecule has its own unique physical properties. Those physical properties involve how they attract other nearby molecules. In the subject of chemistry, there's an adage called likes attract likes. Likes attract likes. And that means that molecules that have similar sizes, similar shapes, attract other similar molecules. And the attraction between nearby molecules, intermolecular forces, sometimes abbreviated IMF, the attraction between nearby molecules is a result of those molecules having similar types of attractions. Likes attract likes. Another way of saying that is likes dissolve likes. That means in order for two things to mix well together, the molecules have to have similar forces. Now, those forces we get into much, much later, but for now, they're forces that are called polar attractions and nonpolar attractions. Polar attractions, and we've already learned something about covalent bonds. Covalent bonds are what holds atoms together in the molecule. But in a covalent bond, there can be two types of attractions, essentially. Polar bonds or covalent bonds are actually made up of two different kinds of bonds. They both involve sharing. If the atoms share the electrons equally, it's said to be a nonpolar bond. If the atoms share the electrons unequally, where one atom is hogging the electrons of another atom, it gives rise to a polar bond. A pole results. The atom that is holding the electrons more, the atom that is hogging the electrons are negative, that atom acts as though it has a negative charge. The atom that is having its electrons hogged away from it ends up with what we call a partial positive charge. Molecules that have a partial positive and a partial negative due to the unequal sharing of electrons results in the formation of a polar covalent bond. So molecules that have polar covalent bonds attract other molecules that have polar covalent bonds. Atoms that are sharing equally have a non-polar covalent bond. There's not a little positive pole and a negative pole. They tend to attract other atoms that have non-polar. Likes attract likes, or likes dissolve likes. In this lab, they're going to use those different properties of intermolecular attraction to separate the colors contained in the pigments in plant leaves. That's the process called chromatography. This is a simulation. In the chromatography lab, you will notice that there will be different parts. The very first page of your lab report is the theory of the lab. Question 1 through 5 is dealing with the theory of the lab. When you click onto the website, this is the website where the simulation will be taking place. To find the theory, click on the Theory tab, and the questions on that first page, 1 through 5, will be answered right here. Right here. Here they're showing you the real process of chromatography. They've taken a substance that is a mixture, and they put it on a piece of paper. Then they're going to take that piece of paper and put it into a solution or solvent. That solvent will go up the paper. That paper will absorb the solvent right up, and that solvent will travel up the paper by capillary action. When that solvent gets to this mixture, the solvent will try to pull that mixture up the paper. As it's pulling it up the paper, the different components in that mixture, the different colors that were in that mixture, will have different properties. The mixtures that are most attracted to the solvent, the parts of that mixture that have intermolecular forces most similar to the solvent, will go up the paper the greatest distance. Because if it's attracted to the solvent will pull it up the paper. Other parts of that mixture, here's our original mixture, the other substances in that mixture may have lesser attractions to the solvent, so they won't get pulled up the paper quite as far. So it looks like we have a color right here. That's the solute. That's the mixture. That solute is the mixture they're trying to pull apart. We have the paper. The paper is often called the stationary phase because the paper is really not moving. You put the paper in the solvent and the paper sits there. The solvent will get pulled up the paper. That's often called the mobile phase because that solvent is moving or is mobile up the paper. The parts of the mixture that are most attracted to the solvent will go up the paper the furthest. The parts of the mixture that are not as attracted won't go up as far. So blue, that light blue, appears to be a color that was in that original mixture. It must be most attracted to the paper. Green, if that is green, is attracted somewhere between. It's attracted to the solvent and the paper. The red appears to be most attracted to the solvent because it was pulled up the paper the greatest distance. That's how chromatography works. This particular color was made up of these three mixtures. So here we go. On the first page of your lab report, the questions are answered right here until you get to this part. So now we are on the portion of the first page which is asking you to look at the video. There's a video on the lab. And what this video is going to do is tell you the other factors that are involved in separating molecules in the process of chromatography. Overridingly, one of the big influences or properties that will allow a mixture to be separated in the process of chromatography is the attraction of portions of the mixture to the paper and attractions of portions of that mixture to the solvent. So that attraction, the likes attract likes or likes dissolve likes, are two of the factors. In this video, they'll discuss other factors. And you'll find that video, you'll find the video right on the tab right there marked video. On the next page of the lab report, you'll actually begin the lab simulation. So now we're going to begin the lab. The purpose again is to use the process of chromatography to identify pigments found in the leaves. And what you are going to do is you'll have an opportunity to change the parameters. There's different pigments that you can select, and there's two different solvents that you can work with. Which ones you will set up are told to you right here on the table. So you're going to do 4 trials. On this one, you can certainly do more than 4. But the 4 trials that we're going to base this lab report are on the effect of ether acetone solvent separating spinach. Then you're going to look at the spinach being separated by a different solvent, the isopropyl alcohol. And then the other factors we're going to compare is we're going to be looking at maple leaves that are around in the spring and summer being separated by ether acetone solvent, and maple leaves in the fall or autumn and winter being separated by ether acetone. Let's take a look on how to set those parameters. So we're now back here on the lab. We're going to click on the simulator. We'll open the simulator. And here on this part of the simulator is where you can mess with which substances you're dealing with. We click here. We have a choice between spinach green, the maple leaves in the spring, summer, the maple leaves in the autumn, winter, and other fall foliage. Now in this lab, we're not using the fall foliage, but you can mess around with this on your own. Here's where the solvents are, the ether acetone and the isopropyl alcohol. So on our very first one, we're going to do spinach and ether acetone. So right here is where we'll start the experiment. I click it, and there's where they have the spot. And here's the solvent going up. And as the solvent is going up through this test tube, we can see that this particular green spinach or spinach green is being separated into the different colors or the different pigments that make up that leaf. And here it is now being separated. The lab instructs you to click on the test tube so we can see it better. So I clicked on the test tube and we can now see it better. And you don't actually have to draw this on the lab report. It's already been copied for you. Let's take a look. So here we are. That's the part that was copied off the lab, off the simulation. That's the spinach green and ether acetone that we saw being separated. And from here, we want to see how they got the grid on. So if you click over here, show scale, show chart, we can see now where the numbers came from. And we need these numbers, we need these measurements to calculate the RF value. It said for us to identify this band, and it's pointing to this band right here. In order to identify this band, I need the RF value. The RF value is how far the color traveled up the paper divided by how far the solvent went up the paper. It looks like the solvent went up about 6.2 centimeters, and this band traveled somewhere around, if we're looking at that spot right there, that the main part of that spot looks to be right in here, somewhere around 2.8, 2.9. So the distance of the pigment is 2.8, 2.9, and the solvent went about 6.2. And we will need to write those two numbers right here because they want us to identify the band. We're back on the lab report. We come down here, and they want the RF value for the one that the band identified. So I would put like 2.8 on top of this. That's the distance the pigment traveled, divided by the distance the solvent traveled, which was 6.2. I divide those two, and that's right around 0.45 or so. And now they want the name of that color. It looks to be chlorophyll B. This chart right here, when it says show the chart, I took the 2.8 divided by 6.2. That was right around 0.45. That's the dark green color, chlorophyll B. To check to see if I'm right, I'll come right over here. I'll scroll down, select the pigment as chlorophyll B. Correct. That's telling me I did that part right. And you'll do this for the other three trials as well. On our lab report, the second trial has spinach green in isopropyl alcohol. So I'll leave the spinach green right here. I'll come down to here and change it to isopropyl alcohol, and then I will start the separation. There's the spot of the spinach green. The solvent's carrying the colors up, and it's going to separate those colors. Here's the colors separated. Then I will click on the test tube so that I can see them better, and you'll notice that picture's right there. Again, show the scale, show the chart, and I will have to identify this same band again, it looks like. Notice on this one that they do not have the red one. It says not applicable, so anthocyanin is not one of the choices. This is how we'll proceed with the lab. And you'll do that for all four. Good luck and have fun.